From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on May 29th, 2025. Hello, Micronauts, and welcome once again to our Quality Quorum. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 96th episode of Matters Microbial. As always, I am so very grateful for your listens and views. Thank you for sharing the good microbial word. We are edging slowly but surely toward episode 100. Can you believe it? As you can see here, my wife, Dr. Jennifer Quinn, is exploring glow-in-the-dark pin designs to celebrate that particular occasion. She's creative. As I think about teaching microbiology in the fall for the last time here in Tacoma, here is one of the stickers that my wife designed for me that I hand out in class. I grew up adoring Dr. Seuss, so the microbes you'll know works very well. I mentioned a former micronaut named Mariko, who helped coin the term micronaut. Very artistic. Here is a cartoon that she drew on an exam, if you can believe it, since she was so taken by the microbes in her water bottle. Amazing creativity. Two things have happened because of this podcast that really made me happy recently. First, I exchanged a glow-in-the-dark tardigrade for this Delovibrio plushie that arrived with the kindest thank you notes about this show. Made me a little teary. I guess it's all the dust in here. And just yesterday, I received this amazing email from Frank Nyando at the University of Zambia, telling me that he found the podcast helpful in his own classroom and lab. Thanks, Frank. These two things meant a lot to me. Many thanks. When I was in middle and high school all those years ago, there were very few opportunities to learn real biology. I was fortunate to have some summer school opportunities that helped me, and teachers interested in science fairs, but that was about it. But microbiology and access to microbiology has become so advanced recently, and so many cool questions are out there. In a previous podcast, Dr. Von Cooper discussed his program teaching high school students about microbial evolution. Dr. Seth Bordenstein and Dr. Sarah Bordenstein also do research outreach involving high school students working with the symbiont Wolbachia. In addition, the Tiny Earth program developed by Joe Handelsman to look for antibiotic-producing organisms from soil has also been expanded to high school students with good effect. But I wanted to hear from high school teachers in the trenches bringing new concepts and skills to high school students. Especially given the current climate, science needs excellent PR, and students need opportunities. So I am delighted to introduce Bob Kuhn, biotechnology teacher at the Fulton County Schools Innovation Academy STEM High School in Alpharetta, Georgia. Welcome to the Quality Quorum today, Bob.
Hi, thanks for having me. I'm very excited.
You know, whenever I see you, I think about my own teachers and I think, boy, I needed somebody like you.
I needed somebody like me too. I didn't know what was out there, for sure.
So I think the first question that you kind of posed to me that we talked about is how you became a high school teacher.
Yeah, I didn't set out to become a high school teacher. I actually had a short career as an environmental scientist after graduate school. So I don't have any degrees in biology, to be honest. My two degrees are in geology, but micropaleontology was the actual subdivision of that I studied. The PI that took me on as an undergraduate, I'm not sure. I mean, we both, I don't think, liked geology as much as some of the other geologists in the department because she focused on salt marsh foraminifera. And she had a grant that I could work on as an undergraduate. And so I did that, and I did a little teaching in graduate school like a lot of folks do. But when I left graduate school, I didn't really have too many opportunities to apply, what I had done my thesis on. So I took whatever job I could get. So I became an environmental scientist with the state of Georgia, and which was a great job, but didn't pay a whole lot. So long story short, I'll fast forward. My wife, not my wife at the time, but my girlfriend at the time, she was teaching English in Korea. And when she came back, she told me that she was going to be going to law school at the University of Georgia, which is where we both studied previously. And we had already been apart a year, and I didn't want to do that again for 3 years. So I said, well, maybe I'll change careers because I really enjoyed teaching in graduate school. And I went back and got my certificate to teach, and ended up teaching sixth graders my first year. And then 2 years later, I was in high school. But I like to remind her that I became a teacher out of my love for her because I didn't want to be apart from her. So it's a roundabout way I became a teacher.
I think that that's a wonderful story, and it emphasizes something I want listeners and viewers to remember, is that there are opportunities everywhere, even when you don't necessarily know it. There are doors that are being knocked on that you're not even aware of.
Yes, absolutely. Thinking back now, it's remarkable that I ended up where I am now. And I tell students this every single day because we teach a lot of students that are very, so our school is a STEM magnet school. So we have a lot of hyper-focused students, and they think they know what they want to do, and they think their path is very linear. And my story is a good one because it has been not non-linear. And so I try to use that as an example about, you don't know where you're going to end up, and, you just gotta keep an open mind and go with it. And I'm very happy that where I ended up. I've enjoyed, this is my 28th year I just finished, and I can't say, besides the COVID year, probably the worst year of teaching. Most of my teaching years have been fantastic.
So how did you get involved in doing microbiology in high school? This was not an option for me, of course.
No, and I guess I can kind of explain how I first actually ever encountered a microbe. If in the slide one that I have, besides my picture there in our lab, the organism next to me in the slide is Wolbachia, and story is pretty interesting. I had a 7th grade teacher, and I lived in Manassas, Virginia at the time, and she had a lot of classroom pets, and we could take turns feeding those pets. And it was a community school, so you could walk to school. So I signed up for a week to, feed the gerbils and turtles and everything else. So I walked in one day to the classroom. It was a lab classroom, and she had all these microscopes set up with all these little, glass jars of liquid under the microscopes for us later on in the day. And I said, what are these things? And she did what most good teachers do, is she didn't tell me what they were. She just said, well, why don't you sit down and take a look? Mm-hmm. And I did. And Wolbachia was the first station I sat down at to look through the microscope. And I literally could not believe what I was seeing. Like, it was beautiful for one thing. It was something I'd never seen before. And then I kind of asked her, like, where did this come from? And she had mentioned, like, pond water has Wolbachia in it. And then I started to think, like, wait a second, like, I've swam in ponds and streams and oceans, and like, these things are there. And yeah, so that had me hooked. I mean, it was just a new world,
Well, let me just say really quickly, I hope that we can insert a video here showing everyone the beauties of Wolbachia. Now, I admit it is a eukaryote, And everyone knows there are only 3 eukaryotes I deeply care about, my wife and 2 sons. But I think Volvox is an awfully interesting eukaryote, isn't it?
Yes, and just with the life cycle and everything else that was explained to me at the time, I just had a hard time believing that that could be possible.
Oh, yeah.
So fast forward to graduate school or undergraduate, actually at University of Georgia. I mentioned that my PI had an opportunity for me to work on a research for undergraduates study, and so we looked at salt marsh foraminifera and another microbe, but not a bacterium, but absolutely beautiful as well under the microscope. And so we were trying to look at similarities between the modern environment and environments and rocks that were preserved, marsh environments. We never really made it to the rock part. She had salt marsh foraminifera in her lab that she was able to culture, and my job was to document kind of the biodiversity of the marsh, and especially in the muds where these foraminifera were living. And again, I mean, it was an environment that I hadn't really had a lot of experience with. The Georgia salt marshes are absolutely beautiful. And I became very interested in this microbe for the time I was studying it. And, so I came back to it. And also, just as kind of an aside, I got a chance to take a protozoology class at the University of Georgia. And, I was pretty locked in at that point of how cool these things were, but really didn't come back to microbes until I started teaching biology in high school a whole lot. So there was a time when I was teaching general life science to students. So, I hadn't thought of using microbes at that point and we didn't have the ability really either. So.
Protists are awesome. People, I mean, in all seriousness, it's so sad that there aren't more people teaching courses in fungal biology and protistan biology. I shouldn't say protozoology. But you know what I mean is that, and what I like is they're so charismatic because they're large enough to see with a low-powered microscope, forms vast and various and beautiful. And I'll put a link to some of the interesting worlds that we see with protists. So why is it important to teach microbiology to high school students, Bob?
Well, there's a lot of angles to that question. First of all, I don't think that they have a lot of experience with microbes. A lot of educators are reluctant to do anything with microbes. Sometimes they're not allowed to do anything with microbes. Fortunately, our district is very good about allowing us to do experimentation, and at our school, it's pretty much encouraged. So I think it's important for them to experience microbes, not always not in terms of disease orientation or things like that, but just as an organism they're not familiar with, to learn about them and learn characteristics about them. And then secondly, I would say I try to teach. Well, first of all, I teach biotechnology all day. That's my job. I teach an intro class, an advanced class, and then a research class. And then I also have a club called DNA Club, which is an independent research program after school. And through all of those things, I kind of divide the courses into molecular and microbiology. And so they get a pretty healthy dose of microbiology for 3 years if they're with me all 3 years. And so our school highly encouraged us to do problem-based learning. So I've been able to frame for the 2 microbiology classes through storylines. I've been able to teach microbes through the lens of antibiotic resistance, which is incredibly topical right now. And we can discuss this more later, but that we do this through Tiny Earth as sophomores in high school. And then we have a phage hunting program as well that we do in their junior year. And those combined, we come at the idea of antibiotic resistance from two directions. So there, I think it's really important because of that phenomenon. And biotechnology in our school is connected to healthcare. We have career tech pathways, and it falls into healthcare. So it's very easy to relate microbes back to healthcare. Not always just with disease either, but this idea of the holobiont and looking at relationships between
Mhm.
hosts and their microbes, very important. So try to come at it from that angle a little bit too.
Well, as I often say, first evolved, last extinct. That's the microbial world. It's also the very basis of our biosphere. And you're absolutely right that we sometimes focus on the small numbers of organisms that cause disease. I call them devil microbes. And you'll see this in the media all the time. And that's one reason that what you do is so important. Now, on the other hand, there are people that take this more positive view, angel microbes, that they will, this yogurt will cure depression, right? Which just makes me roll my eyes. I don't know what to say about that. But I think that the important thing is most microbes don't really care, but they've been here before. They'll be here after. They also lend themselves to great student questions. And I'm so delighted your high school supports this.
Oh yeah, incredibly supportive. Sometimes I think it's because they don't really know what we do in biotech. Like, you know, it's kind of
Yeah.
complicated and a lot of kind of sophisticated kind of tools and techniques we can use. So, but no, my principal is very willing to let me experiment and have students do things where the outcome is really not the focus. In fact, I have a lot of friends here at school that are in the English department, so I'm influenced heavily. So in slide 3, there is something I have on the board every year, the doer alone learns from Nietzsche. And that's really important to me
Hmm.
in teaching, is that I want kids doing science. I want kids trying things and taking risks and not worrying about the outcome. Sometimes they get focused on the outcome. The outcome for a lot of scientists is publishing and things like that. But, that's down the road for maybe some of them, maybe not. But I do want them to do things just because they're learning and they're trying. And we don't really ever talk about what the outcome needs to be. The grading portion of it can be, not always based on the product. So uh,
Right.
that experience is really important to me
And
as they go.
I also want to emphasize to listeners and viewers, I mean, so many times, and this also happens in introductory biology in colleges, there are a series of demonstrations that one does. And those kind of bore the instructors and they certainly bore the students. So the idea, and they're called cure experiences, where you're actually doing an open-ended kind of series of experiments, so much more productive where possible. It's not always possible. And in addition, some folks, I'm not pointing fingers, I'm not being judgy. What they will do is they'll actually use that as an adjunct to their research program. But I'm more concerned with what the students learn. And you bring up the tiny earth business. And I believe you have a photograph of an organism you found that certainly inhibits the heck out of Bacillus subtilis, right? What
Yeah,
slide is that?
in slide 10,
Yeah.
you can see this is from this year, actually. This is the
Hmm.
best microbe we had that was producing something that was inhibiting subtilis. But in fact, this one inhibited everything we threw at it. So
Yep.
that was great.
And what I'm trying to bring up with this is the look on their faces when they isolated it and no one has seen what it does in a challenge experiment like that. And, you can tie that into your curriculum with regard to whether or not we have available antibiotics. So they feel what they do has value.
Absolutely. This group in the slide picture, that it's not the group that's next to the plate that discovered this one, but they were very proud to name this one Gladiator because they felt like it took on whatever came at it. Um, yeah, and so first of all, these are sophomores in high school. So these are kids that are about 15. Um, I don't know that Tiny Earth has a lot of sophomores
Right.
participating, but these kids absolutely love it. And we do it for about 6 weeks.
Hmm.
And every single day they come into the lab, super excited about doing things. And in the beginning, we do a series of experiments leading up to this where they learn, things, the basics like streak plating and
Hmm.
culturing, and so they have some experience in basic microbiology up to this point, but they're still not great at it, and they make a lot of mistakes. Like for example, we have them use McFarland standards to when they make their lawns to try to get consistent lawns,
Oh.
and that took a lot of work because they'd never done anything like that before.
Hmm.
Um, and you can kind of see in slide 10, the group that's next to the plate picture. I mean, that's basically what it's like every single day. That a lot of kids in the lab, a lot going on. Um, but really, at some point, to be honest, I become a facilitator in the room. They
Yes.
are able to work on their own, and I'm walking around helping them, bringing them things, answering questions. It is pretty remarkable for their age, and they're all in on it. So it's great for me.
This is a reminder of the old science fiction writer Isaac Asimov's thing. And I'm going to, because I've quoted it several times in the podcast, I'll just cut it to the punchline, is that science progresses when someone looks at something and says, that's weird. And it's really true. So it's one thing, for example, for someone to have a model of a bacteriophage, right? This is a 3D printed model of a T even bacteriophage. It's actually quite a nice one. But as you say, when the students are isolating their own bacteriophages, I presume from soil, and you give them access, I presume at the university, to an electron microscope, and they actually see what's there.
Absolutely. So we started our phage discovery program 5 years ago. And I have to give all the credit to this to a scientist at Montana Tech, Marissa Padula, who had been doing this in high school and middle school with teachers across the state in Montana, various levels of lab, tools and things like that. She basically organized the program for the state. I was interested in doing this 5 years ago, but C-phages is pretty much geared mostly towards college. So I was
Yeah.
looking for a way to still do it. And she really mentored me through this whole process. So I give her all the credit. But when we started this, if in slide 6, you'll see my first group of phage hunters, just 4 kids. They're all graduated at this point, but we started off not knowing what we were doing, stumbling through it together. This was
Hmm.
actually, at the time, this was an after school program. So they did this all on their own time. So they found one phage initially, which they named Starbird, and eventually found 4 more. So we had some confidence after this. So my second group, which you see in slide 7, um, they're going to be seniors next year, but they started when they were sophomores. They found a phage as well. They named Twig. All of these kids, University of Georgia, Georgia Microscopy Center was incredibly generous. They said, we'll prep your phages for free. Bring the kids up. We want them to see what they found. And so after we got our titers high enough, we sent the samples off and we made the trip over to Athens. And these kids were blown away by being able to see something that
Yeah.
maybe had never been seen before. And in fact, if you on slide 7 on the right, those kids are actually driving the TEM. So the folks at the center said, "Hey, why don't you all sit down and look through your sample?" And
Hmm.
it was an incredible experience. So we're incorporating the phage shunting program to the advanced biotech class, which will be my juniors. And so next year we should have hopefully more that we'll be submitting. And all of these phages are in the library now. at the University of Pittsburgh, in through Phages DB. So as far as I know, all of them are. So that output, and being able to contribute to something, whether it's Tiny Earth or the phage hunting program, I mean, that's a really big component for the students, that they're doing something to contribute to science in
Hmm.
some way.
No,
Um,
I really agree. And by the way, for listeners and viewers, the C-Phages program at Pittsburgh is a really interesting one. Now they have avenues all over the world. As stated by Bob, it's primarily been focused on college-age students. And I don't know if you know this, Bob, it's my favorite story about C-Phages, is they had to institute a rule one year that no one could name their bacteriophages that they found after Nicolas Cage.
yes. Yes, and we had some Taylor Swift references and things like that we had to pull back on, because the kids really, and so that, yeah, we encountered the Nicolas Cage phage thing as well. So
Well, it's cute though, and I genuinely mean that. One of the problems in life, and you get to see it before it's beaten out of students, in essence, is enthusiasm is so important. And we've like moved away from it's bad to be enthusiastic. That's not true. Look how enthusiastic people get about sports. This is the same sort of idea. And the look on their faces that I see when they see something that no one saw before, is just amazing. Bob, when I was in graduate school and I was working with bacteriophages against Cynorhizobium melilodi, I once found a phage that made concentric rings in the plaques. I mean, geometrical looking. And when I finished getting my PhD and I left, it turns out that got thrown away during a move. But I always wondered what that phage looked like. But you see, here's the deal. You're able to do that with high school students.
yeah, it's pretty remarkable. I never thought that I'd be doing this for sure. So, and I don't know how much poetry you've had on the podcast before, but we do have kind of like a poem kind of mantra kind of thing we do in our lab, which is on slide 4, which is from Pete Hine. And as you can see in the poem, the point of the poem is that, the whole point of this is to try things and then eventually make less and less errors. So you won't ever make you won't decrease the errors to 0. But the idea here, the spirit behind what I'm trying to do with these kids is to get them to try and really not worry about outcomes a lot, just gain experience, gain confidence. And so far I think it's kind of worked. So
Ownership, and as you mentioned, and you've used this word yourself, iteration is really important. And some people don't like it when a colleague of mine years ago said it's the in research. But anyone who's done any research for any amount of time has had some wonderful result that they can't replicate. And sad though that is, if it can't be replicated, it's not real.
Yeah, and so the stakes there are way higher than what we deal with, and I also don't want students to be thinking about those pressures, because a lot of them actually
Right.
do. Like, I've had kids tell me, in order to be whatever I want to be down the line, I have to be a first author on a paper before I graduate from high school. And I have to really nuance that, because there are forces that I know, or I might not know, that are driving them towards that thinking. And, and I have to be real careful with my response and to try to guide them a little bit, because I don't want them to lose the idea of the process and finding things out. That's really important. Not to say, I mean, we do have some students that have done some things and published some stuff, but
Mhm.
that's when that happens, that's great. And we rally around that sometimes, but that's never the goal of what we're doing. So,
No, the goal is to learn new things. And I have never done anything in my career that had cosmic value to anyone. but I've uncovered a lot of really interesting things. And the exciting part for me is that was there like a present, for millions of years more. And then I was the first to actually see it, or certainly to have reported it in my case. And is it important? No, but it's part of the puzzle that is the universe.
Yeah, absolutely. And I mean, I think so. I think some of the best science sometimes happens when nobody's looking, or
Yes.
and
Yeah.
there's an intrinsic worth to that you get that doesn't, if it's not published, I understand the pressure of publishing in the real science
Sure.
world and all of that. I'm not naive to that, but there's probably a lot of science that happens in labs every single day that's really cool that I would not like to think it gets lost just because it can't be in a paper somewhere, So
If I had a student that had been through your high school program that came to my university as a first year student, knowing the kinds of skills that they learned from you, can you not see well, of course you can see the incredible leg up that they would have over other students. They'd be able to jump right in because they have the skill set. I'm not unhappy teaching people new skills. That's really my job. But I am just so impressed with the skill set that you and teachers like you have been giving these students.
Yeah, and I have to say, I do have an advantage being at a STEM-focused school. We don't have sports, we don't have, the arts as classes here. We are able to focus in on STEM, directly. And the reason that, I'm blessed with the lab I have and the stuff I have, but that money is appropriated towards this, not to other things. And so it's not it's not a typical high school classroom by any means, but I'm going to run with it because I have it and I can help the students learn how to do these things. And we have had a few students go on to college and get lab jobs and end up getting research for undergraduate projects because of some of the skills they learned here. And that's very meaningful to me as a teacher, knowing that, the things they were doing actually paid off for them in some way as well.
So let's talk a little bit. You've had several microbially themed projects that you've discussed already. The Tiny Earth program, where you're having students look for microbes, I presume from the soil?
Yes.
That have some kind of antimicrobial activity, which we've talked about before. I've done it. It is a real game changer for many students, especially students who think they don't like science, by the way, in my experience. So that's one thing you've done. And a second thing you've done is something I can't do because I don't have access to a TEM. But when they isolate bacteriophages, what is your indicator bacterium for this?
Mycobacterium smegmatis. So
right. So
keeping it simple.
Right. So that's the organism that's used a lot by the C-phages people. And the reason for listeners and viewers it's used is though it itself is not a disease, is not officially a disease-causing organism, it is related to Mycobacterium tuberculosis. So it's a great model system for that purpose. And it grows relatively slowly, at least it does for me. And that makes the phage plaques a little easier to see. And it's wonderful for students to see those things. It's just fabulous. And then you also are doing some work with the Wolbachia project, and I think can you spend a few minutes talking about that?
Yes, I can't say enough how fortunate I was to run into the Bordensteins and get to know them and their lab. They mentored me so much on the molecular and microbial side. And so the Wolbachia project was one of the first things we did when we started our DNA club about 8 years ago. I had just gotten a small PCR machine, so I was able to do more. And this was at my previous school, not here. And it was a game changer for me to be able to offer projects that kids could work on after school. And so we started off with just the general Wolbachia project, surveying arthropods for Wolbachia. And that has increased over the years to some, projects that have focused on specific organisms. And so right now we do a lot of Wolbachia work with pill bugs. So slide 14 shows two of the projects. On the right, so this is actually a project that did get published in the Journal of Emerging Investigators. This was inspired by a French scientist, Richard Cordeau, who works on pill bugs. And we were looking at the Wolbachia influence in pill bugs. Tends to turn males into females in terms of the phenotype, and they actually develop into fully functioning females, but chromosomally are still male. And that we did that with nine students over 3 years in 2 schools, and through the pandemic, we were able to continue that project. And so that paper was a big achievement for us because of all of those outside forces that were working with us. So that was great. And then the current project we have with pill bugs is surveying the United States for these, what they call sex ratio disorders, which Wolbachia is one. And then there's a nuclear insert that occurs from called the F element that is inserted into the nuclear material and can cause feminization as well. So whenever I'm on vacation, or I've actually crowdsourced this, had teachers and educators and just the public send me pill bugs, and so far I think we're on 12 or 13 states, including Hawaii. So we're working on finishing that, and that's been a great project. It's been really fun, and the students really like this concept that a microbe can actually affect a eukaryotic organism in this way. So it's been fantastic.
No, we've had Sarah Bordenstein and Seth Bordenstein on the podcast, and they're fabulous educators as well as top-notch research scientists and thinkers. And one of the things that I used to teach a course on symbiosis, and students were fascinated by the idea of things like parasites or other entities affecting behavior and the development of other organisms. And this is before The Last of Us came on television, okay? Let's be clear. But the whole idea of Wolbachia doing this, and I believe it was Ed Yong who referred to Wolbachia as the most successful pandemic on the planet. These are bacteria, again, that are endosymbionts, and they have a variety of effects on their hosts. This is one of them, but they generally modify the hosts because eggs carry the symbionts and sperm do not. So they want to end up as many offspring as possible. And it's fascinating beyond belief. And there is not one Wolbachia. That's the take-home lesson. There are so many different types. That's a niche that got exploited. And I mean, I raise isopods for some of my beasts in labs, so I can send you those anytime. I've got like 4 species. So nice. I'm getting a little carried away. And by the way, for listeners and viewers, when I was trying to do some DNA isolations, it turns out that Bob here has published a report on how best to isolate chromosomal DNA from isopods, pill bugs.
Yeah, I mean, pill bugs are a great model because they're this species of pill bug is the most common. It's everywhere. Pill bugs are somewhat easy to work with. Sometimes they're tough with PCR to get good data sometimes because they do tend to have some inhibitors, but they're really fun and they don't bite, and they're not incredibly easy. I mean, some people would say, who know more about me than culturing them, I've I've never been able to get them to reproduce in captivity, which is probably more about me than about them. But yeah, they're great to work with. One interesting thing that came out of this is that for the small sample size that we did have from Hawaii, we had like 50 A. vulgare pill bugs from one location, no Wolbachia. So, some folks I've talked to have mentioned kind of like an island effect, perhaps. So, I don't know. That's something interesting. I'd love to be able to get my school to pay for me to go back to Hawaii so I could do some more sampling. But that was on a family vacation. So, but yeah, one other thing with Wolbachia that is coming down the line for us next year, we'll be working with a researcher from Lehigh University on starting to look at local fruit fly populations and seeing what the Wolbachia distribution and type is in those local fruit fly populations. And I have a bunch of students that are very excited about doing that because they've never done that kind of stuff. So
it's a wonderful thing. And I was really struck by what you were saying a few moments ago about the drive for publication. And I'm not in an R01 institution. My goal has been to teach students how to be scientists and to think like scientists, even if they, I do that for the people that come through my lab that don't become scientists. They still think like a scientist when I'm done. And it's so useful to them. I've always viewed that as being really primary. And in addition, the idea of ownership, iteration, as you've mentioned, all of these things are super important skills. And in the process of teaching them the skills, you give them skills that could help them in the employment market as well. So I don't know how many wins that is in a row, but it's a lot of wins.
So I was going to showcase a couple other things too, because
please,
this idea of iteration is very important. So slide 13 has a neat experiment that some students did 2 years ago. So we were kind of inspired by the long-term evolution experiment that had been going on for, I guess, 30 years, something like that, with Richard Lenski. And so we were curious about trying to do this over a short period of time. We already knew of an experiment that was published in the American Biology Teacher, which used a strain of E. coli that tended to mutate a little quicker than the normal K12 we would use. And so that experiment was well known, but we got our hands on some of these what are called ecoplates from the company Biolog, and they have preloaded metabolites in wells in the plate. And so the students were directionally selecting towards antibiotic resistance on purpose over a course of 2 weeks. And then they were pulling samples periodically whenever they did a new culture, and they would put them on the eco plates to see if the metabolite profile changed. And this was really fun for them because they have a full load of classes during the day, so they got a really good crash course in how you can't miss a day and how you have to come in and change your cultures and it took some learning because they would come in and everything would be non-usable and I'd have to say, well, y'all didn't come in yesterday. So 14 days straight for them was tough, but once they got going, they took it very seriously and completed their experiment, and they were able to get data that they could look at, and they were very interested in these different profiles. I don't think that anything revolutionary came out of this in terms of, specific things that they could hang their hat on. They say cause and effect kind of things, but the ability to be able to do this, culture microbes and select them, and then also to be able to see what the microbes were using in terms of their growth had a huge effect on them. Like they had never thought of this like this before, and that data like this could be derived from a very short experiment. So that was really fun. And I liked seeing them progress in that because I think they thought it was going to be really easy, and it turned out not to be. So
So there's a saying I have is that the hand of Darwin is on all that lives. And one of the really awesome things about bacteria is that the ones that are rapidly growing, you can do experiments much more easily with them than with other organisms. And so you mentioned Rich Lenski's wonderful work on the long-term E. coli evolution long-term experiment, which has finally been passed off to other people as well, and is just I get very enthusiastic about it because Rich Lenski is the trailblazer, and those are awfully big microbial shoes to fill for those folks following up on it. But I've met some of them, and they're awesome as well. The reason I bring this up is you're surely familiar with Von Cooper's Evolving STEM program.
yeah,
And one of the powers of Evolving STEM is that for about $80 a pop, if you have a reference genome, and you can get genomic DNA from the final product of your experimental evolution program, for about $80, they will tell you what genes have changed. And so, for example, in your increasing antibiotic resistance, you could do that very easily. I've tried to do that with things bacteria that swim faster, for example. These are questions that we could now give to students, I would say undergrad, I would argue high school.
yeah. His program is fantastic, and I think he actually even works with middle school kids there.
Mm-hmm.
I did have one student 2 years ago order one of their kits for the biofilms and work with that in a different way, and they were incredibly supportive.
Yeah.
But I actually didn't know about the genomic part, so that's good for me to know. That's not a big price point. So
I've done it. I mean, and that's the part that's so cool about it, and the ability to. I wish I could be in the room when your students do this, and then you can sit down and say, okay, here's the reference genome of E. coli to begin with, and here's a hyper swimmer. What do you think has changed to make them swim so much faster? And they have guesses. And then what happens is you show them what they did. And such power.
It is. Similar along those lines, one thing we've been able to do in the last 2 years is, so we were able to get a nanopore mini ion here now. And so one thing that's opened up microbially is to do diversity profiles for different experiments. So two I'll showcase on slide 16, which were really fun this year. One was an honors research project, and one was a biotech research project. I had some students who were really interested in whether or not vegetable type would change the profile of microbes when making kimchi. So they set up a bunch of kimchi cultures in our lab and let it run, and then they took their samples and they used the nanopore to do 16S metabarcoding and see the diversity profiles. And similarly, I had some students that were interested in cheese microbiomes, and so they looked at two cheeses and just compare. They didn't have a lot of time to do multiple cheeses, but they did two, and they were able to find some neat things about that as well. So that the ability for us to be able to do that metabarcoding has really opened a lot of doors. A lot of kids are now starting to think about microbial diversity and where they can find it, and then start thinking about simple variables that they can test. So that's been really great.
I am so impressed because that's one of my goals, and it's a little outside my budget if I'm paying out of pocket because of the way that the MinION works in general. So at some point we need to talk about that, because what a wonderful idea. By the way, I did a little cheese microbiology with students, and when you try and grow those things, I will tell you it is the weirdest bad smell that you've ever experienced. And I'm a microbiologist. I know bad smells, and it smells like the cheese, but something awful with it.
Yeah, they didn't try to plate
Yeah.
anything. I encouraged them, but being
Yeah.
seniors, they were less motivated than
Mm-mm.
they might be. So,
It,
um,
it, it was the one time I had students say, Doc, could we not do this experiment ever again? I said, sure. Anyway, I'm so excited about what you're doing. So we're starting to run a little bit short on time, and that's okay. I want to know what the future looks like for you, because look at the number of things you're talking about today that high school students are doing. And whoever is funding what your students are doing, we need to list them so we can all send them thank you notes.
well, so besides our district funding, the biggest for all labs, I suspect the biggest you know monetary expenditure is reagents and consumables. And when you start moving beyond like kits and things like that, yeah, eventually you can kind of build up, an inventory and freeze microbes so you don't have to buy them or beg for them or
Mm-hmm.
whatever. But, a lot of the for sure, the nanopore sequencing is not cheap. And even, some of the reagents and stuff we use for other experiments. And so, and the other thing too is I never know what the kids are going to want to do from a year to year base. So they can come to me and say, I'd like to try this. And we got to sit down, and in the research classes, they have to actually give me a budget, and they have to actually price things out, and they have
Mm-hmm.
to make a proposal. And so their budgets are small. They have from $75 to $150 to spend on their projects. That's not including things we already have in the lab that they could
Sure.
use because we just have it. So I try to make it so that the school can fund projects so everybody has a chance. And then, from there, it constrains them sometimes to be thinking about simple variables that they can manipulate, because a lot of times they want to do things that are their learning gaps or their ability gaps are too big, and it would end up with frustration for them, and also probably waste some money as well. So part of my job is to kind of hone them in on a doable project that they could get some results from so they can learn how to do research. Again, it's not really outcome driven, it's experience driven. So
Mm-hmm.
the student and myself, we have to come to an agreement once the proposal is approved about what they're going to do. And sometimes that takes a while because, sometimes they're really focused on what they want to do. And, if they can't bridge those two gaps, then we have to kind of pare it down a little bit.
No, I think this is so impressive, Bob. Seriously.
Well, thanks. I mean, it's a lot of fun. To be honest with you, it's in my 28 years, the last 5 years, I've never worked as hard, but we have, it definitely keeps me thinking and keeps me from becoming stagnant. It keeps me wanting to do more things and learn more things.
Mm-hmm.
Since I don't have really a biology degree, a lot of this stuff has been self-taught or through
Yeah.
mentorship from other people. And, that has been a great experience for me, because sometimes it's like I'm still in school. I'm still learning things. One of the reasons I'm here over the summer is I've got things I'm trying out, and to be ready for the fall. It gives me some time to do some of my own experimentation to see what we could do with kids. And that's great for me,
Mm-hmm.
personally. It's very fulfilling.
Absolutely. And one thing it sounds like I'm making a joke and I'm not. Microbiology is truly about community. We like to think about single colonies and pure cultures, but in nature, it's about communities. And the field of microbiology is about communities. And I've said before, I'm not in an R01 institution. And yeah, there's a hierarchy. I call it feeding at the different levels of the research trophic web. But what I want to say about this is that I've been made to feel welcome by the vast majority of microbiologists. and you have two, and that's
Absolutely.
says, there's a saying, it's a big tent. We have lots of ideas under one tent. So I guess for us, it's a big petri dish.
Yeah, I would say it on both directions of that. I hardly ever have higher ed folks say no to me. They're usually very interested in working with somebody in high school because it's not something that maybe is in their wheelhouse or something that they usually do. And just the communication and knowledge I gain from them is indispensable. And then on the other side of it, I don't know how many secondary educators will watch this, but I hope they will. A lot of them will. But I think on the other side of it too is to not be afraid to reach out. And PIs, it depends sometimes on their time and what they have coming at them. But like I said, very rarely have I had somebody just flat out say, I don't want to talk to you, I'm not interested. It's usually much more nurturing than that. So I am incredibly grateful to those who I've been able to learn from. So, because it does trickle down, like they may not understand it, but it does trickle down to the students, and they have impacted indirectly hundreds of my students, and that email or that phone call or meeting at a conference or something, that seems like a very brief kind of thing, but the impact is tremendous. Like you were saying earlier at the beginning, when we started, in these times we're in right now, it's incredibly important for that bridge to happen between secondary ed and higher ed, because we can't afford to lose science knowledge. There has to be a pipeline between those two things.
And science is for everyone. Is the field of science perfect? No, of course not. Nothing human is. But what I would say is that the efforts that you're putting in here are the things that have all kinds of benefits down the line, not even for necessarily someone who wants to do something science related for a career. They're able to look at things more critically because of their experience in science.
Absolutely. Who knows? They may at some point in their lives have a sick parent or a child that with something they don't understand. And if they can think back to logically solve a problem or reflect back on something they learned that helps them, understand a little bit better, I mean, that's really important. So I think about that a lot, actually. The long-term impact of, 10 or 15 years from now when they're older,
Absolutely. Do you have some final advice? I know with the current climate that we've mentioned obliquely about funding situations for other STEM educators in secondary schools?
Well, so sometimes educators have an opportunity through their parent-teacher organizations. A lot of them will do mini grants per year, where you can apply for money. I would say always do that. It's very surprising to me how few teachers actually do that. The other thing is very important, just like it is in higher ed, is grants. There are grants out there for even small ones for research. I've gotten small grants through the Entomological Society of America. One place I've been funded twice now was experiment.com is an incredible resource for funding. It's you're not always going to get it, but it does give you an opportunity to both get your stuff out there and have people crowdsource specifically for experiments, which is different than like a GoFundMe or something, because you're competing with all kinds of things there. But for experiment.com, it's all science experimentation. So that's been great. And then, to be honest with you, a lot of times when I do professional development, they'll offer stipends and things like that. And those go directly back into my club. I usually, that's money I never had. So I just put it right into the club and use that to help fund things. So have there been years where, especially for my club, where I self-funded the projects? Absolutely. And I know not everybody can do that, but in order to do some things that we wanted to do, I was willing to do that. So there aren't any easy answers when it comes to that funding idea. But if you're diligent, you can find money out there. A lot of people don't apply for small grants. So if you're willing to put in some time and work it up, one last thing you can try to do at your school is sometimes you have through the parent-teacher organization, you might have parents that are very skilled at writing grants as part of their job. And if you can convince them to help you learn how to write grants, or to actually take that on as a volunteer opportunity for you to give them a grant you're interested in, and they know the magic on how to write those things, that's a game changer if you can find that, because that takes the burden off of the teacher a little bit, and you already have that resource as a parent in your organization. So that would be one thing I would suggest as well.
That's great advice. Thank you. It has been one of the more inspirational podcasts talking with you here today, and I hope that we can stay in touch and continue to talk about things. Things are always busy for everyone, you and me both, but this has been a wonderful experience for me, and I hope for listeners and viewers as well. So I want to thank you for your time and wish you and your family the very best.
Thank you, Hughes. Well, I really appreciate being asked to be on, and it's been a great experience for me.
Thanks so much. This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode with, as usual, epic links can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Bob Kuhn can be found at the Fulton County Schools Innovation Academy STEM High School in Alpharetta, Georgia. Many thanks, as always, to David Renata for superb editing and Reber Clark for that wonderfully quirky music. I hope that you've all enjoyed being part of our Quality Quorum for today. See you next time on Matters Microbial.